Today we will do an overview of basic immunology. These are a series of lectures which give an overview of all aspects of basic immunology. The topics generally cover antigen presentation, antigen receptor and accessory cell molecules, T and B cell development, regulation of the immune system, and immune tolerance. The cells of the immune system comprise of many cells, including monocyte, macrophages, dendritic cells, lymphocytes, among others. These cells, they are predominantly present in lymphoid structures like the lymph node, where the immunological reaction primarily occurs. Antigens are comprised of either intracellular or extracellular antigens. Intracellular antigens are presented by the antigen-presenting cell on the surface of the antigen-presenting cell. The extracellular antigens are presented by a unique non-overlapping pathway and also presented on the surface of antigen-presenting cells. The process of antigen presentation for MHC class I molecules is initiated intracellularly, where the intracellular protein is first degraded by the proteasome. The peptides end at the endoplasmic reticulum and get loaded onto the MHC class I molecule to be presented on the surface. These antigen peptides are recognized by CD8 T cells specifically. MHC class II molecules present extracellular antigens, which are taken up by antigen-presenting cells. They bind and the peptides are loaded onto the surface of MHC class II molecules in an organelle, which is the fusion of the endosome and the lysosome. And these are presented on the cell surface and recognized by CD4 positive T cells. B cells express immunoglobulin molecules, which are present on the cell surface and recognize antigen directly. The immunoglobulin molecule is expressed and secreted by B cells. The MHC class I molecule presents the peptide, which is then recognized by the T cell receptor on CD4 cells and on CD8 cells. And MHC class II molecules are presented by antigen-presenting cells and recognized by the T cell receptor on CD4 cells. The consequence of antigen presentation, which results in MHC class I and class II presentation, results in activation of CD8 and CD4 cells. B cells bind antigen directly and secrete immunoglobulin. Non-classical antigens, such as lipids and glycolipids, are presented by structures such as CD1, which activate NK cells, and NK cells are also involved in the regulation of the immune response. The immunoglobulin molecule, the diversity of the immunoglobulin molecule is initiated at the germline, where the recombination event takes place, where both the heavy and the light chains are developed through recombination processes and form the variable domain of the immunoglobulin molecule, which is represented to recognize antigens directly. The consequence of the immune responses when antigen is presented to CD4 cells is CD4 cells differentiate into various different cell types, including—
I couldn't find that in your music.
Th17 cells, Th1 cells, Th2 cells, and T follicular helper cells. These cells differentiate and help B cells secrete antibody, and they also help CD4 cells activate CD8 cytotoxic T cells, which then can kill MHC class I expressing cells. At least three switches are necessary to activate T cells in the periphery. They start with antigen presentation, which delivers signal 1 to T cells. Signal 2 is delivered by co-stimulatory molecules such as CD80 and CD86. T regulatory cell effects are induced through that stimulation, which then ultimately results in activation of the T cells into its effector function. Thus, when a resting T cell interacts with antigen-presenting cells and the antigen, it delivers signal transductions, which are mediated through phosphorylation events, which recruit intracellular phosphorylated events and activate the T cell by activating various transcription factors, which are then important in secreting cytokines which regulate the immune response. Overall, the immune response is highly regulated by various cell types which comprise of the immune system. CD8 T cell killing occurs through specific mechanisms such as granzymes and perforins, where CD8 cells secrete these proteins and can kill infected cells. In order to shut the immune response down after an immune response has occurred, a series of events takes place, which is shown in this animation. Antigen is presented, delivers signal 1. Signal 2 is delivered through CD28. After the regulatory T cell has played a role, you can observe a full signal. After the signal is activated, a fourth molecule comes up on the cell surface called CTLA4, which binds to CD80 and CD86 with a higher affinity and delivers signal 3, which is an off signal, and shuts off the immune response. This off signal, which is mediated by molecules such as CTLA4 and PD1, have been studied extensively in the last decade and have revolutionized the development of immune responses against tumors and have created a new field of immunotherapy of cancer. Several molecules in the interface of the antigen-presenting cell and the T cell are being studied to understand this biology. And finally, this course takes the process of understanding the immune response as it relates to development of vaccines, such as HPV, rotavirus, and HIV, and biologics, such as monoclonal antibodies to various cell surface targets, to either autoimmune disease and immuno-oncology. Thank you.